Okay, good morning, student. We are going to discuss about today that is excretory product under the eliminations. Okay, please continuously watching this video because if you in between any interpretation is there or intervention is there, you cannot understand the concept of the structure of the kidney and the urine formations. Next, first of all, animals usually some of that waste material is produced from the body due to the metabolisms. Okay, some of the animals is produced ammonia, urea, uric acid, carbon dioxide. Some of the water also. This is produced by metabolism or excess ingestions also. Okay. Next, you see that this excretory material or waste material completely removed from body or partially removed from the body. Okay. These waste material divided into some categories. First one is a nitrogenous waste, that is ammonia, urea, and uric acid. This is three items is called as a nitrogenous waste. In among three, that ammonia is the most toxic form of that waste that is required to the large amount of the water for excretions. Okay, and another uric acid is a least toxic waste that is eliminated by some small amount of the water. Okay, next, based on this based on this waste material, the animals is divided into three categories. Okay, first one is ammonotourism. Those animals is excrete the ammonia as a excretory product, that is called as a ammonotourism. Okay, this example is a bony fishes, aquatic amphibians, and aquatic insects also. Okay, next one, ammonia is easily soluble, readily soluble. This is excreted by diffusion method through surface of the animals or gill surface of the animals. Okay, as ammonium ions, through ammonium ions. Okay, the ammonia is soluble with some other material and converted into the ammonium ions. Okay, this kidney is not play is important role in the ammonotourism. Okay, you note down, not kidney is not play to the any role to the ammonotourisms. Next, ureotelism. Ureotelism means some of the terrestrial animals, okay, excrete the urea and uric acid using the less amount of the water to conserving the water. This is type of the animals is called as a ureotelisms. Mammals and terrestrial amphibians and marine fishes, okay, they were excrete the urea. That is called as a ureotelic animals. Okay, that is already you know that ammonia is produced by the metabolism process that will converted into the urea in the liver. Okay, this is released into the blood. This after urea is filtered by the excreted by the kidney. Okay, some amount of the urea may be retained in the kidney matrix. Okay, these animals for usage of the osmolarity, osmosis, that some small amount of the pressure is maintained inside of the kidney, that is necessity for the living of the animal, that is called as a osmolarity. Next, we will see uriciotelism, another one animals. These another one type of that waste material excreted type. One is a reptile, another birds, and land snail also. Some of the insect also excreted some uric acid. This one type type of the waste material, nitrogenous waste material. Okay, this is formed into the pellet or paste like structure. Some pellet means small amount or paste like structure. Some you see in that some birds are excrete pellet like structure. That is need for small amount of the water. Okay, this is also called as a uricotelic animals. Okay, ammonotelism, ureotelism, and uricotelicism. Three types of that method is there in the animals. And next, some these materials or waste material excreted some through structure. The structure is different. First one is a protonephridia or flame cells that is present in the platyhelminthes, that flatworms. Examples are planaria and rotifer, some of the annelids and the cephalochordate. Examples are ambiaxis. Okay, what is the primary function? Primarily concerned with the ionic and fluid volume regulation. Example is a osmoregulation, maintained to the osmoregulation. You note out this table also. And another next one is a nephridia. This is tubular in structure that is present in the earthworms and annelids. Okay. Next, this is main function is then remove the nitrogenous waste and maintain the fluid and ionic balance in the body of the animals. Next, another one is the Malpighian tubules. The Malpighian tubules, most of the present in the insect, including cockroaches also. Okay, mainly cockroaches. This is useless for the removal of the nitrogenous waste and maintain for the osmoregulations. And another antrenal glands are green glands that is present in the crustacean like prawns. Okay, that are mainly doing the excretory functions. So note down some of the questions may be coming from here. Protonephridia, nephridia, Malpighian tubules, and the antrenal glands are green glands. Next, we'll discuss going to the human excretory systems. Okay, see the human excretory system consists of that of four items. One is a pair of kidneys and one pair of urethras. Okay, you see here this is pair of the kidneys. Both are kidneys and another pair of ureters. This tube-like structure, na, this is called as a ureters, and another urinary bladder. This is urinary bladder, and another one is a urethra. Okay, this is called as a urethra. Next, first we divided into the external and internal structure of the kidney. The kidney is a reddish brown color, bean-shaped structure. Okay, that is present in the lower level of the last thoracic cavity. Thoracic cavity means your above the abdomen cavity. The lungs is present in thoracic cavity only. And the third lumbar vertebrae is close to the tarsal interval of the abdominal cavity. You note down last thoracic area and the third lumbar vertebrae. This is position of the kidney is present. Next, that is a measurement. You see that length is 10 to 12 centimeter and the width is 5 to 7 centimeter and the thickness is 2 to 3 centimeter. Okay, weight is 120 to 170 gram. Okay, note down. This is also some of the questions is coming from this area also. Next, internal structure of the kidney. Okay, that internal structure of the kidney, you see that is the first one is a hilum. Hilum is inner concave. This area is called as a hilum area. Through this area only some renal artery and renal vein ureter also goes inside and comes out of from that hilum area only. Okay. Next is hilum area. Another funnel shaped area is there inner side of that kidney. That is called as a renal pelvis area. Renal pelvis. Okay. No doubt this is also called as a renal pelvis. Okay. With the projection called as a calyx or calyces. Calyx is a singular, calyces is a plural. Okay, this is also calyces. See in your diagram, is calyces is there. Next comes to the internal structure of the kidney. That internal structure is covered by one of the capsule. Okay, one layer that is called as a capsule here area, and another. Inside the kidney is divided into two regions. One is a cortex region and another one is a medulla region. This is cortex region, another one is a medulla regions. Okay, the medulla is divided into few conical masses that is called as a, this is the red color region. This is also called as a medullary pyramid region. This is present in between the calyx or calyces. Okay, the cortex is extended between the medullary region. That is called renal columns or columns pettini. See, renal columns or columns pettini. Next it comes to the nephrons. Okay, each kidney has a 1 million complex tubular structure that is called as a nephrons. Okay, the total structure is called as a nephron. The nephron is functional units of the kidney. Okay, basic functional unit of the kidney. Each nephron have a two structure. One is a glomerular structure. This is called as a glomerulus, and another one is a renal tubules. Okay, the tubules is divided into two proximal convoluted tubule and a distal convoluted tubule also. That you will see next slide. Okay, first one taken to the glomerulus. Glomerulus is a some tuft-like or some cup-like structure. Okay, that is formed by afferent arteriole. See, afferent arteriole. That is a carry the blood from different parts of the body to glomerulus, and another efferent arterioles that is also carry the blood to the away from the glomerulus. Okay, that's a two afferent arteriole and efferent arteriole. Both are consistent and form to the glomerular structures. Okay, this structure is carried by some of the cup-like structure that is called as a Bowman's capsule. Bowman's capsule. Inside of the Bowman capsule only present to the glomerulus. Next, we comes to the Malpighian body or renal capsule. Okay, Glomerulus is along with the Bowman capsule. Okay, this is Bowman capsule. Inside of that, this arteriole and the afferent arteriole, that is called as a glomerulus. Both are combined is called as a Malpighian body or renal capsule. Okay, next we'll come to the tubules. Okay, first after that glomerulus or Malpighian tubule, some coiled structure is present, that is called as a proximal convoluted tubule. Okay, PCD. And another something hairpin like structure is present. Okay, hairpin shaped. This is called as a Henley's loop. This loop is divided into two. One is a descending Henley's tubule and ascending Henley's tubules. Okay, both. This is also that ascending tubules is continued and formed another tubule, that is called as a distant convoluted tubule or DCT. Okay, this is DCT is open into the some another tubule, that is called as a collecting duct. Okay, this is open into the renal pelvis area through medullary pyramids of the calyces. Next, the position of the parts of the kidney. Where is position? You note down, this is also some of the important questions coming from this area also. One is some the Malpighian corpuscles and the proximal convoluted tubule and the distal convoluted tubule. Okay, the three items is present in the cortex of the kidney. In the medulla region, loop of the Henle. Loop of the Henle means both ascending and descending loop also. Next, the types of the nephrons. Okay, the nephrons, two types. One is a cortical nephron, another is juxtamedullary nephrons. Okay, this is majority nephrons is present in the lobe of the Henle. Both are present in lobe of the Henle only. This is very short. Okay, very short, extended into the medulla regions also. This is called as a cortical nephrons. This side, the juxtamedullary nephrons, this is some of the nephrons. Very less. Okay, this is long and runs off into the deep into the medulla also. Okay, this is two type, cortical nephron and juxtamedullary nephrons. Next, Vasa recta. Vasa recta means the efferent arterioles. This is coming from the glomerulus. Okay, form fine capillary structures. Okay, this is called as a peritubular capillaries. Peritubular capillaries. Afferent arterioles carry the blood away from the glomerulus, that is formed some network of that capillary. Okay, this is called as a peritubular capillaries. Okay, and another, the minute vessel of this network non-parallel to the Henle's loops. Some of these capillaries formed around Henle's loops, some of the network, that is called as a Vasa recta. Okay, this Vasa recta is absent or reduced in that cortical region or cortex region. Mostly this is present in the medulla regions only, Vasa recta. Next, urine formations. How to form the urine? Okay. Next, you see the urine formation is done by three stages. One is a glomerular filtration, and the second one is a reabsorption, and the third one is a secretions. Okay, this is also happened in different parts of the areas of nephrons. Okay, first one is a glomerular filtrations. It is a first step of the blood. It's a filtrate from the blood that is carried by or done by the glomerular structure. Okay, because this is called as a glomerular filtration. Okay, averagely 1100 to 1200 ml of blood is filtered by kidney per minute. Okay, roughly 1/4 of the total blood is pumped by the ventricle region in the heart per minute. Okay, the glomerular capillary is a pressure caused by the filtration through three layers. One is a endothelium of glomerular blood vessels. Second one is a epithelium of the Bowman capsule. Okay, third one is a basement membrane between these two layers. Okay, endothelium and epithelium. Endothelium of the glomerular and epithelium of the Bowman capsules. Okay, is a note down three layer present used for glomerular filtration or across the glomerular filtrations. Next, another one step in the glomerular filtrations also ultrafiltrations. What is meaning of the ultrafiltration? Okay, the epithelial cells of the Bowman capsule called as a photocytes. Photocytes. Okay, this is also photocytes. See in your diagram. Okay, it's arranged some of the manner. Okay, leave some spaces. Okay, in between minute spaces. These spaces is called as a filtration slits or slit pores. Okay, the blood is filtered through these pores or slits. Okay, all constitution are part of the plasma except proteins also filtered through the Bowman's capsule or flow through the filtration sleeves. Okay, because this process is called as a ultrafiltrations. Next, glomerular filtration rate. What is meaning glomerular filtration rate? One of the definition is given. The amount of the filtrate formed by the kidney per minute. Okay, per minute, that is called as a glomerular filtration rate, GFR normally. GFR is a healthy individual is approximately 120 by ml per minute. Per day means is taken 180 liters. 180 liters, no doubt. Okay, next. That is a regulation. If usually function is done by some of the organisms, that is also regulated by the some of the organisms also. Okay, organ also. See, here the kidney have built-in mechanism for regulations. Okay, such mechanism, such organ is called as a juxtaglomerular apparatus and shortly called JGA. Okay, see. Next, what is the functions of this JGA? JGA is a special sensitive region formed by cellular modification of the distal convoluted tubule and the afferent arteriole. Okay, both. This is just a modification of the DCT and the afferent arteriole. Okay, how is work? You see, if you anybody area that the glomerular filtration rate is reduced, immediately the juxtaglomerular cells is present in the distal convoluted tubule or afferent tubule that release the one of the hormone that is a renin. Okay, which is immediately the glomerular flood flow is increased in that glomerulus area. Automatically, that glomerular filtrate rate is comes to the normal. Okay, this is function that's involve one of the hormone that is called as a renin. Next, second stage is a reabsorption. The reabsorptions usually per day, how many liters? 180 liters is produced from the body through the filtrations process. Okay, but urine is released 1.5 liters per day only. Okay, because the 99% of the filtrate is again reabsorbed by the body. This process is called as a reabsorptions. Okay, this process is occurred in different region of the nephron by active process or passive mechanisms. Okay, from some examples, glucose, amino acid, Na plus, etc. They are also reabsorbed through active process. So, and reabsorption water also done by passive. Some another nitrogenous waste material also reabsorbed by the passive process only. Note down, those are things are absorbed by actively, those are things are absorbed by the passively. Okay. Next, this is a diagram books given. See proximal convoluted tubule. What are the things is reabsorbed and a distal convoluted tubule. See NaCl, H2O, HCO3 and K plus H plus. Here H plus and NH3 also. Note down here H2O descending lymph up here. Here NaCl also reabsorbed. And another see NaCl here urea is reabsorbed. H2 also reabsorbed in collecting duct. Note down. This is based on diagram also. Some of the questions will be rising. Next, last stage is secretions. During urine formation, some of the tubular cells present in that nephron that secrete some of the substance H plus, K plus and ammonia for the into the filtrate. Okay, this is used for formation of the urine that will helpful for ionic and acid balance of the body fluids. Okay, this is last stage of the urine formation. Okay, thank you student for your watching. Please share, subscribe our channel, comment about the teaching method and share to your friends. Okay, thank you.